value-added course. After yesterday's class, we have discussed about the protein data bank, and also we have seen other aspects which are related to proteomics. Is my screen sharing?
Yes, ma'am.
Yeah, so I didn't change the date, so just excuse me for that. So yesterday Dr. Kamala had given a brief idea about how to work with the protein structures and how to predict the primary structure, secondary structure, tertiary structure, homology modeling. All this we have been going through for the past 2 sessions regarding the proteomics. So today's class, we are going to look at another important aspect of the proteins, which is related to the mutations which happen in the proteins. So here the mutations, actually when we say mutations in the protein structure, we all understand that there are several types of genetic diseases are there, and proteins are generally coded by the triplet codon. The amino acids are there, and there is a always a chance that one of the codons of the triplet code is being replaced by another amino acid, which can another base, which can be at the first position, second position, or the third position, depending on the position of the change. There will be a replacement of the amino acid. So what we mean by the mutation? So going back to our earlier classes where we have discussed about the alignment of sequences. We have seen how the nucleotide sequences are aligned. We have also seen what is the clustral alignment of sequences, where in the comparison sequence you can find that some amino acids are conserved, some amino acids are replaced by similar group of amino acids, or there is a total replacement of the amino acids. So this plays a very crucial role in many aspects related to the protein function, always as well as the structure. So when we are looking at the proteins, the most important aspect which needs to be discussed is what is called as what is the percentage of accepted mutations. What we mean by this PAM matrix. So here again we go back to the algorithms, or we go back to the calculations based on what are the amino acids which can be replaced, and what are the replacements which can be tolerated by our cells, which amino acid mutations are not tolerated by the cells, whether they are deleterious mutations. So if any of the participants are there from the genetics background, or even otherwise, we classify the mutations under different categories: frame shift mutations, and then nonsense mutations and point mutations. Like this, there are different types of mutations. Each of them is having a specific change which is occurring in the sequence of the amino acids, which are contributing or which are there to the protein structure. So here, after going through the several classes related to protein structure and protein function, we have come to understanding that amino acid sequence plays a very crucial role in deciding the predicting the secondary structure of the protein. And in a previous class, we were also explained about the disordered regions and the Ramachandran plot, which is very important. What are the amino acids which will be coming in the range of the Ramachandran plot? Which are the allowed regions? Which are the disallowed regions? So much of explanation has been given to this. So today we are going to see what is the impact of the mutations, and that is one aspect of the proteins which is important, whether the mutations are going to really impact the structure of a protein. Then the next thing, what is the role of these proteins is, we are also using the proteins for the molecular docking, which you are going to see in the subsequent classes. Dr. Vanitha ma'am is going to tell you about autodock and other parameters where we are trying to take the structure of a protein and dock it with some chemical, so that which is useful for the drug discovery. So many applications are there when we know the protein structure. So what would happen if these proteins undergo the mutations, and how to identify these mutations, and that is one part of the talk. And the next part is going to be looking at the energy minimization of the protein structure. So we'll come to that later. So the percentage accepted mutations, or is a matrix which is made in order to make us understand what could be the amount of new, amino acids which can undergo the permitted mutations, and what are the replacement amino acids for each different 20 amino acids, what could be the most suitable amino acids that they can be replaced, so that we call that as the accepted point mutations. So this replacement of one amino acid in a protein by another residue is has been accepted by natural selection. So when I say that, what I would like to tell here is that when when we look at the genetic code, we know that the genetic code is a degenerate code where there is each some of the amino acids have more than one codon, and even though there are three codons, the same amino acid is being coded because there is a single amino acid difference in the codon. So that is an accepted mutation, that is at the third position. So but if it happens at the first or second position, then it will be a different kind of impact. So this accepted point mutation is what is generally talked about as a point accepted mutations, or percentage of accepted mutations. These are different terminologies given to the same thing. You can abbreviate accepted point mutation, or sometimes they are referring this as percentage of accepted mutations, and so on. So an amino acid change which is accepted by natural selection, this occurs when a gene undergoes a DNA mutation such that it encodes a different amino acid, and the entire species adopts the change as the predominant form of the protein. Okay. So now you can see here, this is the beta globin, delta globin. If you can recollect the class on phylogenetic tree construction, which we had seen, the globins are a set of proteins which can be classified in different types, or they can be taken up in different tree constructions, where you have the plant globins, the animal globins, all these are categorized in different manners. So the beta globins, the delta globins, alpha globins, and myoglobins. Example is given here. It is indicated that if it is, they are all changing with respect to a particular amino acid mutation is here. They are replaced with one particular position. You can see that here the beta and delta globin have almost the same sequence, except this, but alpha 1 globin, myoglobin, and other types of globins, they have a replacement of a particular type of amino acid. So what is A, what is G, what is E? What is the type of nature of these amino acids? Anybody? Yes. What is amino acid? Letter A stands for? Hello? Participants? No participants?
You can use a chat box. Someone has written alanine.
Hello? Are you all following?
Please respond.
Okay, so shall I continue? I continue. Yeah. So alanine is being replaced by glycine, or alanine is replaced by another amino acid with this E. What does E stand for? Amino acids E. Audible audition. This sound is not clear. Okay. So you can see that it's a different type of amino acid, and because of that replacement of the amino acid, these two ones, or these two um, have shared a common ancestor, and these are present in a different link altogether. So this kind of replacement of the amino acid is important, as we have seen, for the process of natural selection, how the species are going to be getting differentiated, or how they are going to be grouped into separate categories based on their amino acid sequence. So that is what we have seen regarding the importance of the sequence of amino acids. So today, what is the way we can, what we are going to look at is the mutations, how to identify the mutations in the amino acids, and find out which is a better, which mutation is a deleterious mutation. What are the tools we are going to use to analyze the mutations which are present in a particular protein. Okay, so now, so directly today, I'm going not going much into the details of the theory behind this. Already we have seen these changes of their sequence, and here there is one matrix, as we have seen earlier. This is the PAM matrix, which is called as the point accepted mutations, or the percentage of accepted mutations. So this is a probability matrix which is important, the PAM1 mutation probability matrix. So in this, you have seen there is a amino acids are arranged in the row wise, as well as the these are the rows where all the amino acids are listed, and you have column wise the different amino
Padma's
acids.
slide did not change.
It is
Scoring
showing it is
matrices slide is scoring matrices slide only is seen.
just a
Box is
minute,
not seen.
because it is showing sharing.
Yeah, I didn't change the slide, I think so.
Now you can see
Um,
our matrix.
not
You
it.
can see.
Not it. Okay, I'm sure. Not it, ma'am.
Is the size changing? I didn't.
Stop sharing, you
No,
do, and
ma'am,
again you
it's
try to see, ma'am.
screen is blank, ma'am.
Are they sharing now?
No, ma'am, it is still like that. Totally blank.
Just a minute, I will. Is it sharing?
No, ma'am.
Now?
No, ma'am. It is still like that, blank. Ma'am, just it shared, ma'am. Just 1 second. I was able to see that. Not again.
Is it visible now?
No, ma'am, it is the same. Now it is visible, ma'am.
Oh, it is visible.
Yes, ma'am. Now it is visible.
You can see the slide.
Students, please respond. We can see the first slide, ma'am.
I changed the slide. Is it sharing? PAM matrix.
No, ma'am. It is showing the only the title slide, mutation analysis and energy minimization.
Huh?
Is it sharing now?
Yes, ma'am. We can see the screen,
You know.
but no, the slide is not yet open.
No?
No, ma'am.
No?
No, ma'am.
And for me, it is saying it is sharing. Maybe
Only blank white screen is visible. That's it, ma'am.
it's recording. I'll restart.